for inviting me to speak here today. As you can see in this meeting, maybe someone who's just happy about achieving atomic resolution is not too exciting for you. But what I actually want to talk about is joining mass spectrometry, specifically electrospray ionization mass spectrometry, with electron microscopy or other highly resolving microscopy techniques to create something beyond what we are capable of doing currently. I'm from Oxford, so this is an ancient place. As it is with ancient places, you can show nice buildings and look back on a strong history. And what I'm showing you here is one of the older examples of microscopy. So I'm at Wadham College, which is the college of Robert Hooke, and so most of you will be annoyed in school with Hooke's law and moving on springs, but what many maybe do not know is that many contributions to the current design of light microscopes go back to his time, and from him there is a wonderful book, Micrographia. You can see it open on our librarian's desk when we were allowed to come in and look at these old volumes and see these hand-drawn pictures. And I found it extremely inspiring to see, when you think about it, that at the time, for the first time, people saw details of this flea here, and to think what this means for our research. And it is quite interesting that indeed we find things that trouble us as well. So in the chapter about imaging an ant, you find that Hooke was quite displeased with his progress when he was not able to make it suffer its body to lie quiet in a natural pose. So he struggled with sample preparation as well. And so it would be destroyed, he could not use wax or glue. And in the end, as so often, alcohol came to the rescue. So rectified spirit of wine did the trick. So it would have probably killed the ant, but without destroying it. And he could image a sample and draw this beautiful picture. So you can try it for yourself if you have a light microscope still. I think it is still a wonderful exercise. And so his statement that it is the nature of minute bodies that as soon as almost as ever their life is destroyed, their parts immediately shrivel and lose their beauty, is still an issue that we face today. So our methods are more advanced. I'm coming originally from surface science, solid state physics, where we, for instance, use scanning probe microscopy. And scanning probe microscopy is an interesting case. So the method got the Nobel Prize, interestingly, together with Ernst Ruska for electron microscopy, for basically just showing one atomically resolved image. So the first paper was atomic resolution and Nobel Prize, if you will. Quite an achievement, but you can see it took many years, to 2009 actually, to achieve something similar for a molecule, and it was pentacene. So with a trick to pin a carbon monoxide molecule at the tip using non-contact AFM, people could do atomic resolution in atomic force microscopy, and it is still kind of an art to do this, and very difficult. And so why is this method not used everywhere if you can make images at atomic resolution like this? And the reason that we stumbled into is, of course, sample preparation. So in SPM, we are working with crystals. We have to clean them in ultra-high vacuum in order to make an atomically smooth surface. And then if we want to image molecules, we have to keep this state. Hence, we have to resort to thermal sublimation and vacuum, use a crucible, that's where the molecule is in. So that would be a terbium diphthallocyanine molecule that you then get onto the surface. And of course, it is clear when you then think of organic biological molecules, something more complex, you're stuck. Because these don't have vapor pressure, they are not stable at high temperatures. So with this methodology, we don't have a chance to bring them onto the surface. So what we resorted to was using another method that gives you intact molecular gas phase ions, or gas phase particle, which is electrospray ionization. So you all heard of this, of course. So from a molecule in a solution, You can electrospray, disintegrate these droplets by one of these models, but in any case, you get an intact molecular gas phase ion that you have in an ion beam. And then you can do a lot of things with this ion beam. Specifically, you can mass filter it in a mass-selective ion optics, like this quadrupole here. So for instance, pulling this one peptide ion out from this group here. And you can mass analyze, so measure these spectra. You can accelerate, slow them down, and ultimately land them on such a surface. So this would be your STM imaging workflow, cleaning sample, deposition in ultra-high vacuum, and then imaging. Before I want to show you an example of this, and then also talk about electron microscopy, I want to point out how well controlled this technique is. So I explained to you already that we can pick out a single peak from a mass spectrum within bounds. We then decide where we deposit. So what you see here is an ion beam image of our sample holder. So we have a focus beam, we raster it left and right with deflection plates. We see where the sample positions are, and we place our beam exactly at the sample positions. We can adjust the beam energy, we measure it, and we apply bias voltages to the sample to slow it down to a collision energy as low or high as we like. Sometimes we like high collision energies. Sometimes need to break stuff, but normally we go low. And we can measure the current on the sample. I hope you can see it down here. And current times time is a charge. We know one molecule is 2 charges here. So we know exactly how many molecules are on our sample. So this is how such a machine looks in detail. So if you want to discuss ion optics a little bit, be in touch with me later. I only want to point out, so you see there are several sample holders for several types of application. Basically each sample, each microscope gets its own sample holder, and we take a look at these in a moment. So for STM, just briefly, it works really, really well. So you can deposit proteins, peptides, glycans, synthetic molecules. You can go as far in this one as observing single spin flip events in these. And I very briefly want to show you that by this technique, scanning tunneling microscopy actually becomes a useful technique, because we can use it to image glycans, which are always a little bit a nightmare analytically, because you have things like configuration, connectivity, branching, and quite generally they are very flexible, and it is very hard to image them. It's hard to work with them in mass spectrometry, and hence we tried what we can see in STM. So this is literally taken from the sugar in the coffee corner. So this is sucrose, and if you zoom in, you see the 2 sugar subunits. You don't see this for every disaccharide. So some of them clump together a little bit. So it shows you already that we have to think about conformation. But if we go to bigger glycans, we can usually make out the main chain very well. We can make out branches, even count how big these branches are. And of course, we can apply the mass selectivity. So if we're starting from a mixture, we can pull out one peak or the other. We have 2 samples, take the images, and distinguish that the pentamanose here is, actually it's wrong way around, so that's the pentamanose is linear and the hexamanose is a branched glycan. So my colleague Calvin Angara, who took over my old lab in Stuttgart, is currently pushing this further by looking into glycopeptides and beyond, and so he assembled a whole zoo of things. So glycopeptide, glycosamino glycans, where he imaged sulfonation sites, really big glycopeptides, where he can just by contrast distinguish what type of glycans they have. Glycolipids I find especially impressive, where you see the aliphatic chains in contrast to the glycans. And I think the most beautiful things he can show us is these really extremely long extended proteins, where you can read the glycosylation site more or less directly from the chain. Here for an RNA's N-glycan with a single glycosylation, or for a heavily glycosylated O-glycan here, mucin, where here in these individual images indicated each glycosylation site. Of course, this presents us with a problem, and this leads us here. When we look at these branched glycans, we can see that we cannot read the connectivity directly. So the mannose in here is either linked alpha 1,2 or alpha 1,6, and if we identify them and then measure the bond distances, we see the information is still there, but in the STM image, the information is hidden. So you can see below here, perhaps, the information is hidden in the configuration of the individual sugar subunit on the surface. And so as a final point here, in the beginning I mentioned with non-contact AFM, when you pick up a carbon monoxide molecule to the apex, you can achieve this atomic resolution on these flatline molecules. We tried the same recently with glycans, so with cyclodextrin here together with Ben Mayada and Bruno Della Torre in our midst. And what we used was cyclodextrin, so our first test molecule. And you can see here in STM, you would get the blobs always, right? So you can't tell more detail than that you have 7 sugar subunits or dextrin subunits in here. But once you pick up a carbon monoxide molecule at the tip, you see 2 distinct types of STM images. They correspond to a primary and secondary phase. Cyclodextrin has a conical shape. So there's a narrow phase we call primary, and there's a wide phase which we call secondary. And we can very nicely distinguish these 2 phases. And if we compare them to atomic models, then we can go so far as to identify these features here, which are really now at atomic resolution with different OH groups in the sugar. So you can see the secondary phase has 2 types of OH groups, and it presents you with 2 types of features due to the electrostatic environment of these OH groups. And there, I think we are at a very similar level like in electron microscopy. So we measure some sort of a density. We have an imaging theory, which gives us information at atomic resolution. It is a little bit more convoluted than the imaging theory that underpins electron microscopy, but nevertheless, here we have a technique that at atomic resolution can tell you, in principle, which type of sugar you have. And so maybe this is a good transition to the second part that I wanted to show you about electron microscopy. So here there's no discussion that we're dealing with a method that can deliver atomic resolution. On the material science side, I experienced this myself. So this is a typical reference measurement here, a silicon lattice in 110 direction. So the material scientists show this to flex about their microscopes and say, so you can distinguish these 2 atomic columns at 80 picometers here. This is something we did ourselves with our method. We deposited cesium iodide clusters to look how these magic number clusters look like, and they actually look super interesting, not at all like in the gas phase, so they reorder, but you can clearly see the carbon atoms of the freestanding graphene underneath, and the cesium and iodine atoms adsorbed on the surface. So atomic resolution is very well possible, and I don't have to tell you the same is true on the cryo-EM side, where with averaging we've seen that we reach into the 1 angstrom resolution range nowadays. So the issue I want to talk about is sample preparation. And there is a wonderful working sample preparation pathway through application of liquid blotting and then plunge freezing into liquid ethane that gives us these very thin water films and water ice films. They are amorphous and a little bit lower density, so we get contrast to the embedded proteins. But you all know that many things can go wrong in this sample preparation pathway. And in addition, the preparation to get here, to make a sample of pure protein that you can apply to your plunge freeze and workflow also can be quite tedious. That's why we thought maybe native electrospray ion beam deposition might be a way out or support in some difficult cases, and we wanted to explore what this does. On the other hand, when you think about native mass spectrometry, so here I outline, it's the same mass spectrometry pathway, only you make sure that you choose the right buffers and narrow emitters, very low voltages, so that you treat your protein as gentle as possible. So thinking about this, native mass spectrometry gives us amazing results, like the spectra of these viruses here. This is from Albert Hecht's group, or very complicated assembled complexes that we can reassemble here. And in parallel, we can obtain structures, but how do they really shake hands? How do they fit together? Is what I observe here in mass spectrometry really the same as what I see in cryo-EM or in crystallography when I get structures of these? Because I go very different pathways. On the one side, I ionize. On the other side, I perhaps plunge freeze or crystallize my sample. And I think this is the other interesting question. So what I'm asking here is, first, can we make samples? Can we do this pathway? So especially with a thing that, let's call it native soft landing, and a transfer mechanism, so that we can get structures from particles that were mass selected in the gas phase first, and the other way around. Can we relate structures that we find here to native structures, that how they are in solution? In order to do this, we have to think about what happens on this pathway. So, of course, we have something happening to the protein in the gas phase transfer. So when we have an electrospray and we ionize our particles, we take a lot of the solvent away, maybe all of the solvent away. This certainly will affect our particle. When we fly it through the gas phase, we collide it with other gas molecules, we collide it with the surface, it absorbs on the surface. Also, there are forces working on the particle. And finally, when we handle the sample, so we transfer it through different stages into liquid nitrogen, into the cryo-EM, also this can affect our particles. So to work on this, we built a machine, this time not self-built like the other one for the STM I showed you before, but this time it is based on a UHMR Orbitrap instrument, which we modified after the HCD cell by this ion beam column and a very special landing optics. It can do the same thing what I described before. You have a quadrupole mass filter, you have an ion source, you have a superb Orbitrap analyzer with very high resolution, and you even have other capabilities like a collision cell where you can play around with your ion beam. I explain this in detail a lot later. So just to get it out of the way, so this is how native mass spectra look like on our machine. So a few test things we work with, the usual suspects, ferritin, ProEL, beta-galactosidase. And you would always get your native peaks here. Oh, I hope this one. A bit more motivation. Your native peaks, and then select it down to clean up the spectrum to only deposit what we consider native. So with the initial prototype of our machine, we did a few first attempts. So the structures that you see here, so raw data, 2D reconstruction, and 3D, was done on the first iteration of a prototype where we landed our molecules just on room temperature grids, room temperature amorphous carbon grids, completely free of any ice, and then plunge froze these. So with beta-gal, we saw that we would roughly get the shape of the beta-gal molecule. In parallel, colleagues in the US, here in the group of Josh Kuhn, did something where they landed, in this case here, also beta-gal, but also here the ProEL into a liquid matrix at the end of their mass spectrometer, and also got rough shapes, even with some details of the proteins. So this motivated us on the one side that this works, but it put up all these questions, what we really have to do to get to high resolution structures here. the important point about our instrument was that again we built our ion optics such that we could control all these parameters. I indicated this already for the STM work, and here I want to point it out again. So the first important point is we can be selective here. We pushed it a little bit to the limit of selecting a single charge state here. So it's a single charge state of native BSA, which gives you 30 picoamps of current. So a picoamp is 6 million ions or 6 million charges per second. So you can calculate how much you need. So we measure coverage in picoamp hours. So 1 picoamp for 1 hour is 1 picoamp hour. And it's 10 to the 9 roughly particles or ion charges. And so sufficient density you get at 5 to 10 picoamp hours. So 30 picoamp hours made us very happy at 30 picoamps because it means 20 minutes deposition time. So this is something we can work with. Actually, this was not for free. For this, we quite heavily modified the UHMR interface. You can see normally you have vast losses here. So in the beginning, you measure almost a nanoamp, and then later in the instrument, it's almost immeasurable. Mass spectrometry is very sensitive. We saw this in the talk yesterday, down to the single ion. A single ion on a TEM grid, you don't want to go searching for this one. So we need a bit more. So we optimize the instrument so that we get current in the 100 picoamp ranges, which works very well. What was most crucial is a little bit convoluted in this graph here. These are energy distributions measured, and it's the end of the slide. And don't mind the absolute energies. So we measure versus ground, so sometimes we have negative energies. So don't be bothered by this. It's okay to have negative energies sometimes. And the important point is the width of the energy. The energy width tells you how rough your protein is treated while it flies through your beam column. And so we worked really hard by optimizing our beam conditions to get this width down to 1 eV, 1 electron volt energy width in order to treat the particles gently. Okay, so the next thing which we worked a lot on was the transfer. I already introduced you to the special landing optics and promised to say a little bit more about it. So what you see here is a cold stage. So we want to land on cold TEM grids. They are hidden in this sample holder here, and the sample holder slots in here, and you have a deposition optics in front. So all of this is at temperatures you can go down to 40 Kelvin with a little Stirling cooler. And in the end, you want to go into the liquid nitrogen bath, like here, and take out the samples. So for this, we adapted an Aquila stage transfer suitcase, so we call it suitcase because we carry it around, to our sample holder that can pick up our sample stage from in here, takes it out, so these shutters automatically close. We many times rinse the volumes in between and then plunge it into liquid nitrogen. So this is how we transfer our samples, so they never see atmosphere, water vapor, and the likes. The next thing you have to think about is substrates. So you want to have an electron transparent substrate. So here's a few candidates: graphene oxide, amorphous carbon, and graphene. You see, we picked amorphous carbon. Graphene oxide shows charging. You see, everything wants to land on the rim, not on the sample. Graphene shows diffusion. Everything lumps together in these chains. They are quite beautiful, but we want to have individual particles. So amorphous carbon it is, because it is a little bit sticky. Important surface-induced dissociation, so collision energy. We did these experiments. We didn't have to. We knew that at 2 eV, everything would be fine. I'll show you in a second why. But it is quite important to see we can go up in energy. We can break the molecules. We actually see individual subunits appearing by, I think, the collision mechanism, as I indicated up here. So 1 unit sticks, and the rest flies away. And so the rough shape and assembly of the protein here, beta-gal, is retained. Is the fold retained? So we were quite certain about this because a while before we simulated these collisions for a different protein and a different system, but informative enough. So this is freestanding graphene colliding with a protein here at 5 eV per charge, so it's a relatively high energy, but you can see that this gas phase fold here is retained throughout this collision, and there's actually very interesting dynamics happening, sound waves in the graphene, and I don't go into detail here, but thin membranes are particularly good in taking away energy, as in the old firefighter cloth, where you should jump in from a building. In contrast to this, the high energy collision, luckily much faster, but you can see that there's so much energy transferred into the protein that the fold disassembles, and you can imagine that this protein will maybe arrive as a compact lump, but not as a folded protein. This was 50 eV per charge. So finally, the question is, do we need a matrix? Do we want to put the protein just on the grid, or do we need something around the protein? So normally this would be amorphous ice coming directly from a liquid, a quenched liquid. We cannot do this in vacuum when it is cold already, but what we can do is we can grow ice. And you see we can make different types of samples. So this is a reference sample. This is how this looks like, and you can clearly see the ring in the Fourier transformation for the amorphous ice. If we grow or make our samples by deposition, there's no ice visible here at all, unless we want to. So we leak a little bit of water vapor into the chamber. We wait for a while, and then ice is growing, here in this case polycrystalline ice, but we can also make this entirely amorphous, different morphologies, thickness of layers. Also this can be very well controlled. So now some results here. First an overview. So beta-galactosidase, we pick out these peaks here which correspond to the folded protein, and we deposit it on the surface. We find these very highly resolved 2D classes, and then generate a 3D structure from this. In parallel, we always do this with plunge freezing because we want to have something to compare with the same measurement. So with these structures here, we were able to achieve a resolution of 2.5 angstrom, and from this we were able to build a structure. But you can already see in the rough comparison that once things are similar, so these indications here for alpha helices and beta sheets, we find again from the reference, but these cavities here that are present on the protein surface, they have been gone. And this can be very well seen when you just morph one of the structure into the other. Here I overlay a coarsened structure to 8 angstrom. So this is a model built from the structures we found. So this was the reference structure here. And now I just morph it into the dehydrated, I already said it there, so into the gas phase structure that we observed after deposition. You see the fold is still present, the subunits are still present, but these cavities that were filled with water before it went into the gas phase, these have been closed. So in more detail here, if you hold them next to each other, you can see that the, let's call it gas phase structure for now, or adsorbed structure, is a little bit more compact than the reference structure. And if you run a cross correlation over the structure, you see that the best correlation you find at the inside of the protein, whereas at these tips and on the surface, the correlation gets poorer. In detail, if we look at typical alpha helices and beta sheets, we find very, very good agreement for the secondary structure. So where changes occur are in the arrangements of the domains and subunits. So it's also interesting to look in detail at some interfaces. So some of the interfaces are retained to a very high level. Other interfaces are quite dissimilar. But always you see what is an alpha helix before it also is an alpha helix in our gas phase structure that we deposited. We simulated this entire affair with molecular dynamics. So namely, we took the PDB structure, we solvated and relaxed it, and then we slowly removed desolvation, or actually relatively fast, so simulation time a few nanoseconds. And we end up at something that looks almost exactly like what we observe in electron microscopy. In silico, we can then reverse the process. So if we add water again, the cavity is open and the original fold is restored. It is quite interesting at this stage in the simulation to look how much water we still have. So from the mass spectrum, we were judging that we don't have more than 200 water molecules. So in the simulation at the final stage, what we call dehydrated, there was still 1,400 water molecules, and you see it barely even makes half of a water shell. So hydration really needs a lot of water molecules in order to affect a protein. And so this brings me to a conclusion here. So what did we achieve with this deposition? So we achieved an atomically resolved map, and we could build a structure. We found that secondary structure is largely retained. We found a small difference in the arrangement of the protein in the subunits, that these cavities are closed, and we can clearly pin this to a dehydration. So I give credit to the colleagues in a second still. I want to show you a little bit of an outlook. So we reproduced this many times with more or less the same level of success, each time finding the same types of structures. So this is reproducible. We can do this with other structures now. So this is GROE-L. Also here you see the fold is retained. You see a slight deformation. GROE-L interestingly seems to shrink and expand in another direction. So this also has to do something with mechanical properties. Other proteins are extremely rigid, like C-reactive protein. It's actually really nice to see it upright standing on the surface after landing and ice embedding. And it almost changes no shape as compared to the PDB. Other proteins, again here this GDH, we find very well resolved structures now, but it seems that these little tails here can only be observed in solution, so they are disordered. We don't see their density anymore, for now at least. So the most important, our recent attempts show us that we can also work quite well with membrane proteins. So here we create little shells of surfactant. You can see each of these peaks represent a surfactant molecule still attached to the protein. So we're currently optimizing this a lot. We want to get to a situation like this. So this is our reference from solution, where we get like a lot of surfactant around the protein to stabilize it. But you see already now, in comparison, our 2D classes do not look so bad entirely. And so with this, my big main conclusion, so I think electrospray ion beam deposition for cryo-EM is viable. You do not get exactly the same as in solution, small changes, but I think we can learn things from this. And I think that this is interesting, shows me that the colleagues are building a lot new instruments now. So the Stuttgart instrument is still my machine. Most of them were for the purpose of STM, but as you've seen, also a few colleagues are trying this for electron microscopy. Good. And with this, as promised, a bit of credit. So most of the work in the EM you've seen, I did with my colleague Kim Esser. So he's now with Thermo. So just last week he started there. We are still all very sad. So they don't smile like this anymore. So Tim is gone. He is not gone. He's with Thermo. We collaborate a lot and very fruitfully. It's great fun. I highlight Kelvin here, who did amazing work with the glycans in Stuttgart, and the many colleagues in Cambridge and Oxford that support us here. Thank you very much.